All right, are we ready to finish up genetic engineering tools? Yay! Let's review real quick first on what we've already covered. So with genetic engineering tools, we first talked about, I think, those little additional loops of DNA that you could introduce to bacteria. You could shove them in there in addition to their regular DNA and then trick them into reading that DNA. What do we call those little loops of DNA? Plasmids, okay. We can engineer plasmids by using restriction enzymes to cut them and leaving sticky ends, and then we can use restriction enzymes to cut out the gene that we manufactured and stick it into a plasmid and put that into bacteria, and that's how we can alter bacterial DNA. If we've got time, I will show you an example of that later. We talked about restriction enzymes a little bit, what restriction enzymes do. Restriction enzymes cut DNA at a known sequence. There are several different types of restriction enzymes. Restriction enzymes are quickly being made obsolete for reasons we're going to talk about today. But I wanted you to know what a restriction enzyme is because they're still a commonly used genetic engineering tool, even though their day is numbered here. It's kind of teaching you about old-fashioned technology now. Then we can take restriction enzymes. We can take the same DNA and cut it up into lots of different slices using different restriction enzymes. And then we can run that sliced up DNA through one of these gels. What do we call that? Electrophoresis. Gel electrophoresis is how we're going to sort DNA by size, right? And that gives us clues about where the DNA would be cut. And by getting clues about where the DNA is cut, we can tell where a restriction enzyme gene sequence occurs in the DNA. So this starts giving us clues about the order of the A's, T's, C's, G's in different parts of DNA. Right, and this is how we make people's unique genetic fingerprint. This is how they're doing like crime scene analysis and figuring out that like the DNA left at a crime scene was indeed left by the perpetrator and not by anybody else, is because everybody will have a unique genetic footprint or a unique genetic fingerprint. Why do I keep saying footprint? Have a unique genetic fingerprint when you run their DNA through a gel cut with several different restriction enzymes. Everybody will have some places in their DNA where they get cut where nobody else's DNA would be cut by a restriction enzyme, thus giving you unique patterns when you run gel electrophoresis. Neat? Are we happy with all that so far? So far, I think that's. Oh yeah, if we want to make a gajillion copies of DNA, what do I use? PCR, polymerase chain reaction. That works just by taking advantage of the fact that DNA wants to be a self-replicating molecule. If you give it all the equipment to do self-replication, DNA will merrily replicate itself. So, quick review of how we use all this stuff. So you don't have to write all this down if you don't want to, because again, I'm just reviewing everything that we've used so far. Because this would be where I stopped teaching about this stuff 3 years ago. This was all the biotech you needed to know about because CRISPR-Cas9 really hadn't hit the scene yet. So, imagine you work at a chemical company and you design a drug based on a human protein. So, you design a gene that codes for the order of amino acids in your drug because that's how you built your drug. So, you make a gene that would code that would dial up the right amino acids in the right order. How do you get that many copies of your engineered gene? PCR is how we make copies, right? Think PCR. PCR is the DNA copy machine. All right, great. How about, how do I know if the A's and T's and C's and G's are in the right order? Because I can't see the DNA I just manufactured, so how am I going to figure out where all the A's, T's, C's, G's are located? Ooh, this is the tricky bit. I'm going to cut with lots and lots and lots of restriction enzymes, right? I'm going to try to use enough different restriction enzymes. Because remember, I know where restriction enzymes are going to make their cut. I know the gene sequence where the cut occurs. So if I treat with one and it cuts once and leaves two equal-sized fragments, well, then I know somewhere in the middle, that particular sequence of A's, T's, C's, G's, that's where that's located, right? And if I get enough of that data together, I can piece together what the sequence of A's and T's and C's and G's must be across the entire gene I've engineered. So I'd use restriction enzymes and gel electrophoresis to figure out that information. That is a painstaking, difficult, awful process, but that's how DNA was decoded, at least at the beginning here. That's how they did the entire Human Genome Project. They decoded literally 6 billion base pairs of code by just taking DNA, cutting it with restriction enzymes, looking where the cuts occurred, and then just doing that over and over and over again until they pieced it together. The guys that worked on the Human Genome Project said it was a little bit like taking a whole set of encyclopedias, putting it in a chipper shredder, and then you just get all the fragments out, and then trying to repiece together the entire encyclopedia. Like, that's kind of the daunting challenge that was doing the Human Genome Project. Nowadays, it would be a lot easier. We'll get to that in a minute. Okay, how do you get your gene into a bacteria to trick that bacteria into making your drug for you so that you don't have to do some complicated manufacturing process? You just make a living thing, read your DNA, and crank out the protein you want. How would I do that? How do I introduce DNA into a bacteria? Make a plasmid using restriction enzymes. So I take engineered plasmid, made with restriction enzymes, I introduce it to a bacteria. All you got to do is this trick called heat shock. You heat shock the bacteria, they suck up the plasmid, then they start reading the DNA that you gave them. And they'll just crank out the proteins for you. So that's handy. How do you check to see if your plasmid has the code you want? Again, if ever I'm trying to read DNA code, I'm going to use a bunch of restriction enzymes and run it into gel electrophoresis over and over again until I'm sure that I've got the right A's, T's, C's, G's in the right order. Ta-da. Good? Are we understanding basically how these tools will be used? Again, me explaining how the tools are used is not nearly as good as you guys actually using the tools, which I wish we could do this year, but I have no way to do restriction enzyme labs at home. So, sorry, you're stuck with learning about it rather than doing it, but that's the beauty of this year. Anyway, now let's make all of this sort of obsolete. Not all of it. Some of it's still going to be used, but her invention is going to change the game here. In 2013, that scientist right there, her name is Jennifer Doudna, she figured out how to do CRISPR-Cas9. And she figured it out accidentally by working with microbes that help us make yogurt. She was a yogurt scientist. Now she works at Berkeley and has a Nobel Prize. So nice job, Jennifer. There you go. Founded by, figured out by Jennifer Doudna. Really what CRISPR-Cas9 is, it's a naturally occurring part of a microbial immune system. That's basically what's going on. She worked with food industry, okay? She worked with a yogurt company. This is how she stumbled onto this. When you work with microbes, the microbes are all doing battle just like we are with all their pathogens. You get it? So if I'm a little yogurt-producing fungus, okay, you know how to make yogurt? You want to go over how to make yogurt?
Yeah.
Here we go. Ready? Quick rundown on how yogurt's made. Step one, take cow, milk it into a cup. Yeah? That was me milking cow into a cup. Yeah? So now I got a cup of milk. Yes? Milk is all loaded up with all kinds of milk sugars. Like there's sugar and fat. That's basically what milk is. It's just sugar, fat, liquid. It's modified cow sweat. Ew, gross, but that's how mammals do. Anyway, so I've got a cup of milk, yes? Now I take some little microbes that do lactic acid fermentation when you cut off their oxygen supply. So I take some little microbes, they're going to be yeast, microbes, or bacteria, it doesn't matter, but some single-celled organism that's going to do fermentation. I throw them into the milk and let them merely chomp up the milk. Then I let them eat the milk for a little while, make all kinds of little bacteria babies. Then I cut off their oxygen supply because I put a top on the milk. Yeah? If I cut off their oxygen supply, they're going to start doing lactic acid fermentation because they can't do full respiration anymore. They're going to poop lactic acid as a waste product. That's what gives yogurt its tart taste. It's literally the microbes that lived in the cup that are still alive in there when you eat it. Like they've been doing lactic acid fermentation, and that's what they do. They're good guy bacteria. We like them. They help your guts. What?
So let's say you got yogurt at the store. So the longer you wait to eat it, the more tart it would get. Or no?
Yes. To some extent. They're not terribly active at the store. That's one of the reasons we keep it refrigerated. That's one of the reasons why it would spoil if you just left it on the countertop, tried to eat it a week later. Those bacteria would go gangbusters at room temperature. Like they would just eat every bit of sugar in that yogurt. And what you would get is something really gross, like highly concentrated lactic acid. So don't eat unrefrigerated yogurt. That would be unpleasant. But if you refrigerate it, it slows down all their biochemistry, right? Because at slower temperatures, chemistry goes slower. So they're going so slowly in your refrigerator that they're not doing much noticeable fermentation anymore. Okay, so we got yogurt. We got these microbes that live in it. The microbes that live in it give yogurt its taste, give yogurt all of its properties. Without the microbes, you wouldn't get yogurt. You'd get a gross cup of milk sludge. So you want healthy little microbes that are going to do the lactic acid fermentation. If you work at a yogurt company, these little microbes are your nice pets that are making your product for you that's making you money. Yeah? So you want to keep your nice little pet bacteria nice and happy and not invaded by other viruses and stuff. Because bacteria get sick just like we get sick, which is weird to think, but it's true. Okay? Single-cell microbial stuff is picking up pathogens from its environment. They've got to have a defense system, much like you have this whole defensive immune system. Part of their immune system was this CRISPR-Cas9 mechanism that microbial critters have. She was the first one that actually sat down, her yogurt factory kept getting sick. This is people that work with microbes, anybody that works in like a fermenting type industry. I went to go tour an ethanol plant once. That was, and the scientist that was leading us around the ethanol plant was like, ooh, come over to this fermenter over here, smell how it smells bad. And we'd be like, yeah, he's like, that's because this batch is sick. Like this whole wing of the factory caught a virus and it's just running rampant through the factory. And until we fix that, we're like way down on production. So like that's what a lot of these people do that work in food services or food industry or work with making ethanol, anything that's a product coming out of microbes, which is a lot of stuff that humans use. They worry a lot about their microbes getting sick. So Donna discovers that this mechanism is being used, and she thinks, wait a minute, I could use the same mechanism with a little bit of tweaking. It's a naturally occurring mechanism in these microbes, but I can control in a lab how this thing works, how this naturally occurring system functions. I can modify it a little bit, and I could use it to make cuts to DNA anywhere I want, and I can program it. And that's the kicker here. What she discovered, what CRISPR-Cas9 really is, I'm going to kind of oversimplify it here, but think of it as being a restriction enzyme. You know what restriction enzymes do, right? They cut DNA. It's a restriction enzyme, but instead of just being a restriction enzyme that, like, I buy the restriction enzyme and it's going to cut at a predetermined code because of how the enzyme is shaped, what she discovered was basically a programmable restriction enzyme. In other words, she could write the code where she wanted to make the cut. That's what makes CRISPR-Cas9 different than all the restriction enzymes. And since you're taking advantage of a naturally occurring mechanism anyway, this is part of an immune system of a microbe, it's cheap. It already exists. Like, she just refined the process to like take control of it and reprogram it, but it already existed. It's no more expensive to use CRISPR-Cas9 than it is to use old-fashioned restriction enzymes. So that's pretty cool. Everybody always asks, what does CRISPR stand for? And every time I read it, or every time I talk about it, I have to go and like read it again to make sure I say it right. Which used to make me feel really bad. I'm like, man, I've been teaching about CRISPR-Cas9 for a while here. I should eventually remember the acronym. But then I watched an interview with Jennifer Doudna, and somebody asked her, what does CRISPR stand for? And she had to like pull out a piece of paper. She's like, I need a cheat sheet every time. So then I was like, yes, if you invent it and you still need a cheat sheet, then I don't feel so bad. But it stands for clustered, regularly interspaced, short palindromic repeats. Ugh. That's why they called it CRISPR, because it's a lot easier to say. The name itself gets into some of the how it originally looked when bacteria were using it for a part of their immune system. Turns out that viruses and stuff will have these palindromic repeats. You know what a palindrome is? It's same forward and backward, like race car, right? If you lead race car forward and race car backwards, it's the same word, yes? So many dynamos. Palindrome.
A jar for a can of tea.
Yes. But anyway, these palindromic repeats were sections of DNA that they'd repeat palindromically. So you'd have like AAA, TTT, AAA. That's palindromic, right? Like either way, it reads the same way. That's how bacteria were doing it, and now we're not really limited to these palindromic repeats anymore because we figured out how to reprogram this, but that it came from that historically. And so these palindromic repeats were an important part of doing the location mechanism. You don't really need to know about it, but that's what it stands for in case you care. CRISPR-Cas9 is an evolutionary adaptation found in bacteria. They use it to cut viral DNA out of their own genome, because some viruses, when they invade you, they actually take their DNA and stick it into your DNA. A lot of that junk DNA that we've talked about before in you is stuff that viruses have shoved into the human genome over billions of years. Bacteria were using CRISPR, naturally occurring CRISPR, as a means to snip out stuff that didn't belong in their DNA. Dr. Dowden's lab figured out that you could use the same trick to locate and change DNA in humans. So that's her big breakthrough. She took something that was naturally occurring in bacteria and figured out how to use it in humans. It was her and a French scientist whose name I can't remember, but it was the first Nobel Prize that was awarded to all women, in case you care. But that's That's sort of interesting. They've been doing Nobel Prize for a long time, like since the invention of dynamite. Like,
That was the first Nobel Prize.
yeah, trinitrotoluene was the first Nobel Prize, TNT, like big dynamite.
Fancy.
Yeah, so it was one of the first Nobel Prizes, and obviously they invented that a long time ago. And since then, there has not been an all-female Nobel Prize until CRISPR-Cas9, so that's kind of cool. And the Nobel Prize was awarded for that last year. So this is like recent. Like I'm teaching you stuff that is definitely not in your textbook because it didn't exist when your textbook was printed. Question?
Yeah, so relating to like your third bullet point,
Yeah.
I was just reading the point of our book, I think it was chapter 18 about, at the end of it, it's talking about like cancer and how like if you get a certain virus or, well, several viruses can actually like, give you cancer.
Yes.
They change in DNA.
Yes.
So like could you use CRISPR to like edit that out if you already have cancer or like how?
Yes. All of the above are true. There's certain types of viruses that when they try to shove their viral DNA into your normal DNA, it screws up your DNA so bad that it gives you the mutations that are necessary to give you cancer. A good example of that would be HPV, human papillomavirus. You've probably heard of that one. HPV does that. It actually can alter your DNA to the point where it gives you cancer. It results in cervical cancer and a staggeringly high number of females that get infected with HPV end up developing cervical cancer, and that's kind of a huge problem. But since we know what it does, like we know where the virus DNA gets inserted, with technology like CRISPR-Cas9, we can go in there and cut that back out, just like how bacteria were using it to cut out viral DNA. So that's coming, that's on the horizon. We got to figure out how to use CRISPR-Cas9 on a whole intact organism. So far, nobody's figured out how to do it for an entire multi-celled organism. They have done it, and they have done it to humans in the embryonic stage. There was a Chinese scientist that used CRISPR-Cas9 to insert a gene into a human embryo to make the babies HIV resistant. They inserted a gene, and these kids presumably grew up to be like full-grown, like he said they made it, full-grown like adult humans, and they have a gene that makes it so that they won't get HIV virus. That's been done. It was super duper controversial and against all the international rules that everybody has agreed on not using this on human embryos yet, because this is brand new technology. Like literally they discovered the existence of CRISPR-Cas9 and that it could be used in this way. They discovered its existence in 2013. It is too fresh of a technology to start using on human embryos. And I agree with that. And Jennifer Doudna, actually, her most recent book was all about, like, I invented this thing, but let's be really freaking careful. We're talking about altering DNA, and not just altering DNA for, like, one person to treat one disease. If you alter germline DNA, the DNA from your reproductive organs, you're going to pass that change on not just to the person that you treat, but all of their progeny from there on out. In other words, it will change our evolutionary path. It's spooky technology, okay? I'm not saying it's good or bad technology because that's impossible, right? Think nuclear bombs. This is like as big a deal technology as understanding how nuclear technology works. With nuclear technology, we can do X-rays and we can do all kinds of medical technology that have like saved millions and millions of lives, or we can build nuclear bombs and kill everyone, right? So like understanding how the nucleus of an atom works, sure, that was a big step forward in science knowledge, but we got to be careful about how we implement that in society, right? And that's the same deal with CRISPR-Cas9. This is like total game changer. It's way too early to just be like, let's go use it on people. We're getting there. We're getting closer every day. They're going to do it soon. The single nucleotide polymorphism stuff, that's coming in the next 5 years in a responsible, well thought out way, I hope. But it's a little too soon to be using it on human embryos. But this Chinese scientist did it. His government was not okay with it. The whole international science community was not okay with him doing that. He got fired. He got disappeared for a while, but I heard he reappeared. He's still alive. At first they thought the Chinese government might have made him not be alive anymore, because it was kind of an embarrassment that like it was a government-funded lab in China, and they dumped an awful lot of money on him and thought he was doing one thing, but really he was trying to use CRISPR on human embryos just to be the first guy that did it. Yeah, it was super ugly. The kids exist. The kids are alive. There are CRISPR-altered humans that exist out there.
When did that happen?
Last year, 2 years ago.
Oh.
Maximum 2 years ago. It was recent.
So the kids are like little.
The kids have been born though. He didn't like report out his research until it was too late to do anything about it. You get what I'm saying? Like he wanted to make sure the kids were all the way to like kids are born. And
Yeah.
then he said like, look what I've done. And the whole rest of the world was like, don't do that yet. What are you doing? Yes.
So, if you were to like take out like the genes that cause cancer from someone that got like it virally,
Yes.
would you still need like the doctors to actually remove the tumors though? Like, I mean.
Yes, if you already got to the point where there were tumors and stuff, then, well, let me think here. If you're not actively able to make more tumor cells because the genes are gone,
It would be like benign.
then your immune system should be able to take care of the tumors.
Oh. So would oncologists kind of be obsolete at that point once you could do it?
Yes and no. I mean, there's always going to be... cancer is the result of the accumulation of mutations in the cell line. It will eventually happen to everybody if you live long enough. So even if we like fix all the... like, here's places where your DNA went wrong, we go in and we edit those and we fix them all, it doesn't mean you couldn't redevelop new mutations that would also lead to cancer later. So I don't think oncologists would ever like be out of work, but their toolbox is about to get a whole lot better. But in kind of a scary way because powerful new technology, like you don't want to screw something up and like wreck our entire genetic path. Like, I don't know, we're literally like taking the reins on evolution and steering it where we want to go. That's scary technology. Like we have to be super cautious.
You could fly.
Her books are really good. I like her. Like, she's kind of funny. She writes at a level where she doesn't insult you by assuming you're too stupid, but she's also not like way up here. Like, she's definitely like biochemist type scientist that's like way the heck up here. And if she wanted to, she could read a book that I wouldn't be able to read. She could write a book I wouldn't be able to read. You get what I'm saying? But she doesn't. She does a nice job of like giving you enough. She's assuming you had high school science, but she's not going to dumb it down below that, and does a really nice job of writing so that normal humans can understand. And every time I see her in an interview or something, I'm like, wow, she seems like smart, but able to talk to normal people. That's rare in scientists. Usually once scientists get to that kind of level, they're like so much into their stuff that they can't believe the rest of the world doesn't understand science to their level. Whatever, that happens. What?
So this is something that's like really cost effective for like what it can accomplish, right? Like,
Yes.
It's just bacteria.
That's one of the reasons why it's such a big deal, is that it's incredibly cost effective. This is cheap technology.
So do you think that like once it gets more like streamlined and able to use in the medical field, it's gonna be something like insulin where they just jack up the price of it for the heck of it?
Hopefully not, but yes, that's a danger. She writes about that in her book too, that like people don't understand that like we're still in the dark as to like, can I patent my particular CRISPR that I made that's going to cut DNA at this part? Like, can I patent that code then?
Basically just patent a restriction enzyme.
Right. It's like, yeah, even though it's not really a restriction enzyme anymore, it's just a sequence on your CRISPR-Cas9 that's already been invented. Can you patent that? And like, all of that is in legal right now. So it's definitely, it's a very cool time. The toolbox for geneticists is about to get much, 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 much better. But it's also complicated because it's such a powerful tool and it's so cheap and affordable that all the ethics of it are going to get very interesting over the next decade or so. CRISPR is the location tool. CRISPR is the part, getting into like what I mean by CRISPR-Cas9. CRISPR is the part that you can program to any sequence in the DNA that you want. And you can program it with a really long sequence. Meaning that, like, I could have a sequence of A's, T's, C's, G's that's a couple thousand base pairs long. A couple thousand base pairs is going to be unique to just one address on the DNA. So with a restriction enzyme, if I just took like a whole human genome and whacked it with a restriction enzyme, that restriction enzyme sequence is probably going to repeat hundreds of times throughout your entire genome. You get what I'm saying? But with CRISPR-Cas9, I can make a long enough sequence that it'll be unique to just one place. It'll cut in only one place. And then around that cut, I know the entire sequence, thousands of base pairs at a time, rather than like look at a restriction enzyme. That one's got like 6. Yeah? And that's pretty typical for a restriction enzyme, that they're looking for a code that's like 6 letters long. With CRISPR-Cas9, I could program a CRISPR to be thousands and thousands of base pairs. So I can read huge chunks of DNA all at once. Speeds up all this stuff with DNA decoding. That it's much, much better at that than the old tools were. Here, watch a video. Here, watch a video without sound because I'm stupid. Hold on. This might be insanely loud. If it is, I'm sorry.
The CRISPR-Cas9 system is a tool for cutting DNA at a specifically targeted location. The technique has already revolutionized gene editing, but scientists are always looking for new possibilities. So what else can CRISPR do? Since being discovered in a bacterial immune system, CRISPR-Cas9 has been adapted into a powerful tool for genomic research. There are two components to the system: a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound
That's the
together,
CRISPR
they
part.
form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes. But making double-strand breaks isn't all CRISPR can do. Some researchers are deactivating one or both of Cas9's cutting domains and fusing new enzymes onto the protein. Cas9 can then be used to transport those enzymes to a specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates specific DNA bases, eventually replacing cytidine with thymidine. This kind of precise gene editing means you could turn a disease-causing mutation into a healthy version of the gene, or introduce a stop codon at a specific place. But it's not all about gene editing. Several labs have been working on ways to use CRISPR to promote gene transcription. They do this by deactivating Cas9 completely, so it can no longer cut DNA. Instead, transcriptional activators are added to the Cas9 by either fusing them directly or via a string of peptides.
Amino acids.
Alternatively, the activators can be recruited to the guide RNA instead. These activators recruit the cell's transcription machinery, bringing RNA polymerase and other factors to the target, and increasing transcription of that gene.
What they just did there, the increase in transcription stuff, was like taking advantage of all that stuff we learned in the first half of this unit.
A CRAB domain fused to the Cas9 inactivates transcription by recruiting more factors that physically block the gene. A more outside-the-box idea for using CRISPR is to attach fluorescent proteins to the complex, so you can see where particular DNA sequences are found in the cell.
Ta-da! Cheap
This
and
is
easy, read
useful
your
for things
DNA.
like visualizing the 3D architecture of the genome, or to paint an entire chromosome and follow its position in the nucleus. CRISPR has already changed the face of research, but these new ideas show that what's been achieved so far could just be the tip of the iceberg when it comes to CRISPR's potential. Whatever comes next, it seems the CRISPR revolution is far from over.
That's from the scientific journal Nature. If you ever see anything in Nature, that's one of the big name journals. Like, that's a big deal. Anyway, so that's all the stuff that CRISPR-Cas9 can do. In other words, it takes most of the older genetic technology and makes it pretty obsolete. Because you can read DNA using CRISPR, you can alter DNA using CRISPR. It's as cheap and easy to use as a restriction enzyme. It's fast and easy. There's kits that are affordable, like at the high school level. And next year, when we have a real school year again, I'm hoping that we can do our first CRISPR labs in here, which is pretty impressive when you consider this tech it's not even a decade old yet. Like, it was discovered in 2013. And we're less than a decade in and we're already using it in a high school classroom. That's pretty impressive. Like, This is going to drastically change the world. I don't know. To me, it's exciting, but also kind of a little terrifying. That's it, really. I don't really want you to know that much about the gene chip or the microarray. Your book mentions it a little bit. It's a technology that's kind of taken a backseat. This was the one that people were excited about before CRISPR hit the scene. CRISPR is going to kind of rewrite how all this works, but this is going to speed up how fast we head into genetic medicine. If you were going to go into like pharmacy work and stuff, it's about to get more complicated because it used to be if somebody had, I don't know, cancer, okay, we would prescribe everybody the same drug, but now what they're doing pretty regularly is screening your particular DNA and then altering your course of treatment at a pharmacy to match with your DNA because reading and understanding DNA just got so much cheaper and easier because of CRISPR. We're entering the age of genetically tailored medicine, and that's a big deal. Total game changer. It's going to make everything in healthcare work a lot better. What's that going to do to the ethics and the cost and the ever-raging healthcare who pays for it debate? I don't know. It's going to make all of that more complicated for sure. But it's also like benefit to humanity is going to be enormous, just absolutely huge. By the time you guys are at an age where you're going to be thinking about like having a family, making kids, whatever, this is going to be something that's available. Like there won't be stuff like cystic fibrosis or hemophilia. Like that's going to go. All the stuff that's caused by like single letter mutation, all the bad diseases that are like one single letter stuff, that's all going to be gone. That's going to be the first round of stuff that gets fixed. I'm excited. I think that's pretty cool. So that's everything with gene tech that I wanted you to know about. Make sure you understand that CRISPR-Cas9 is like super important. It's a big deal. It's like modernizing how restriction enzymes work. Yeah? Are there questions about anything?
Wait, so we don't need to know about the DNA microarray?
I don't need you to know much about the DNA microarray. It's a way to quickly gene screen people. What they're envisioning in the very near future is that you would go to your doctor, you'd get your entire genome screened, because that used to be, it used to cost $7-8 billion to read out the order of your A's, T's, C's, G's. In 2003, it cost about $7 billion to do that. In 2021 now, it costs under $100. That's how fast biotech is going. It went from $7 billion worth of technology when they did the Human Genome Project in 2003. Now, if you want to, you can take a cheek swab, send it off to 23andMe or Ancestry DNA, and they can decode the entire DNA strand, know the order of all your A's, T's, C's, G's, and they can do it for like $100. That's crazy. That's like, it bothers me a little bit that the general public doesn't understand how fast biotech is going right now. It's mind-blowing. This is happening, like, your kids will be reading about the time in history where humans took control of their own genome. Like, it is total game changer. This is like as big as Industrial Revolution and stuff. Everybody is living through it together, and nobody even sees it being a thing yet because the tech hasn't really hit the general public yet. But it's gonna, and it's a total game changer. Question?
So would those companies that like do the genome screening,
Yeah.
would they use CRISPR with
Yes.
the fluorescent proteins to do it?
Yes, now they do. 5 years ago, they were using restriction enzymes, and that's why it was costing so much, is because trying to crunch all the data that came out of using these little, like, 6-letter at a time reading of DNA stuff and then trying to compile all that information just involves a huge amount of computer work and data crunching. We don't need to do any of that anymore. Like, CRISPR-Cas9 made it so cheap and so easy to screen DNA. I don't know. It's an exciting time. It's a little bit of a terrifying time, too, when you actually read about, like, what you can do with it. Figuring out what we should and shouldn't do with it is going to be more complicated than what it can do. The ethics are really going to be the time-limiting factor on how fast we can start fixing kids with cystic fibrosis and stuff. Okay, there are review videos up. I'm going to try to keep the quiz short and sweet. Make sure you understand pre-transcriptional, transcriptional, and post-transcriptional gene expression regulation. That's going to be on your test tomorrow. Quite a bit of it. So pre-transcriptional will be stuff like manipulating histones to make DNA readable or unreadable. Transcriptional will be all the stuff we talked about with promoter regions, enhancer regions, activator proteins, general transcription factors, RNA polymerase, all of that business. You should understand, like, how do I actually activate a gene, turn it on, and make it go. That's transcriptional regulation. Post-transcriptional regulation would be RNA editing. Once we've got the mRNA made from the DNA strand, I can hack out the introns, splice together the exons, and all that business. So manipulating the mRNA, that's post-transcriptional. And then after that, I suppose still post-transcriptional would be things like once I've made the whole protein, I can either activate or inactivate the protein using allosteric inhibitors or active site inhibitors. That's all part of gene expression regulation as well. Were you happy with all that? Is there anything else anybody is worried about? That was a lot of talking, sorry. All right, I'm going to be done with this.